awesome day, my beloved STEM Legend class. How are you today? I am glad you are here joining me in learning today. I am Teacher Ernest, and together we will discover the exciting world of biology. Riding a Ferris wheel will take you up, but return to where it started like a cycle. But before that, let's have a short review of our past lesson about the prokaryotic and the eukaryotic cells. Are you ready? What are the common features or similarities of a prokaryotic and the eukaryotic cells? Very good! The common features or similarities of a prokaryotic and eukaryotic cells are the DNA, the cell or plasma membrane, and the ribosomes. Seems you really understood our past lesson. Another wonderful day for us to learn. How is your day going, dear biologists? In our past lessons, you have learned that the cell theory that cells came from pre-existing cells. Today, we will get deeper into this postulate. You will learn how cells reproduce. At the end of the lesson, you are expected to define cell cycle, describe the phases of the cell cycle and their control points, and differentiate interphase and mitotic phase. Now let us explore the cell cycle and their control points. The cell cycle is an ordered series of events involving the cell growth and cell division that produces two new daughter cells. Cell on the path to cell division proceeds through a series of precisely timed and carefully regulated stages of growth, DNA replication, and division that produces two identical cells. The cell cycle has two major phases, the interphase and the mitotic phase. During the mitotic phase, the replicated DNA and cytoplasmic contents are separated and the cell divides, according to Belardo in 2016. During interphase, the cell undergoes normal growth processes while also preparing for cell division. In order for a cell to move from interphase into the mitotic phase, many internal and external conditions must be met. The three stages of interphase are the G1, S, and G2. Number 1 is the G1 phase or the first gap. In this stage, the cell is accumulating the building blocks of chromosomal DNA and associated proteins as well as accumulating sufficient energy reserves to complete the task of replicating each chromosome in the nucleus. Number 2 is the S phase or the synthesis of the DNA. During the S phase, DNA replication can proceed through the mechanisms that result in the formation of identical pairs of DNA molecules, sister chromatids that are firmly attached to the centromeric region. And number 3 is the G2 phase or the second gap. In this phase, the cell replenishes its energy, stores, and synthesizes proteins necessary for chromosome manipulation. Some cell organelles are duplicated, and the cytoskeleton is dismantled to provide resources for the mitotic phase. The mitotic phase is a multi-step process during which the duplicated chromosomes are aligned, separated, and moved to opposite poles of the cell, and then are divided into two new identical daughter cells. The four stages of mitotic phase are called prophase, metaphase, anaphase, and telophase. Number 1 is the prophase. During this stage, the nuclear envelope starts to dissociate into small vesicles, and the membranous organelles fragment and disperse toward the periphery of the cell. The nucleolus will disappear. The chromosome begins to move to opposite poles of the cell. Microtubules that will form the mitotic spindle extend between the centrosomes, pushing them farther apart as the microtubule fibers lengthen. The sister chromatids begins to coil more tightly with the aid of condensing proteins and become visible under light microscope. Number 2 is the metaphase. During this interphase, all the chromosomes are aligned in a plane called the metaphase plane, midway between the two poles of the cell. The sister chromatids are tightly attached to each other by cohesion proteins. At this time, the chromosomes are maximally condensed.
Number 3 is the anaphase.
At this stage, the cohesion proteins degrade and the sister chromatids separate at the centromere. Each chromatid, now called a chromosome, is pulled rapidly towards the centrosome to which its microtubule is attached. The cell becomes visible elongated as the polar microtubules slide against each other at the metaphase plate where they overlap.
And the number 4 is the telophase.
At this stage, the chromosomes reach the opposite poles and begin to decondense, relaxing into chromatic configuration. The mitotic spindles are depolymerized into tubulin monomers that will be used to assemble cytoskeletal components for each daughter cell. Nuclear envelopes form around the chromosomes and the nucleosomes appear within the nuclear area.
Now you know the phases of the cell cycle and their control points. At this point, let us check if you really understood our lesson through a multiple choice activity. Number 1.
What phase in the cell cycle where the cell undergoes normal growth processes while preparing for cell division? A. Interphase. B. Mitotic phase.
Very good. The answer is interphase. Number 2.
What phase in the cell cycle where G1, S, and G2 phase usually happens? A. Interphase. B. Mitotic phase.
You're right. The answer is interphase. Number 3.
What stage in mitosis when the nucleolus disappears? A. Anaphase. B. Prophase.
Very good. The answer is prophase. Number 4.
What stage in mitosis when the chromosomes are aligned in the center or equatorial plane? A. Anaphase. B. Metaphase.
Yes, the answer is metaphase.
Number 5. What stage in mitosis when the sister chromatids separate at the centromere? A. Anaphase. B. Telophase.
Very good. The answer is anaphase. Congratulations, my dear STEMligent learners. I hope that you learned a lot today. Once again, I am your energetic teacher saying, eagerness to learn will lead you in the fulfillment of your dreams. See you in the next episode.